As mentioned, I'm Matt Spear, Chief Medical Officer at PoseidaTherapeutics. We're a CAR T-cell and gene therapy company with a number of unique platforms and products. Here are my disclaimers. So, CAR T-cells, gene therapy, and a number of unique platforms I'd like to discuss in detail in the next few minutes. We're based in San Diego, approximately 250 employees over in the UTC area. A lot of very smart scientists have joined from local and distant universities to make this come true. So, what are those platforms? First of all, piggyback transposons as a means of gene insertion, which is somewhat unique in the field. It has a number of different advantages. Clover for gene editing. It's a proprietary system we've developed, effectively a hybrid between zinc fingers or talons and CRISPR. And, methods of delivering these platforms, moving from AAV to novel lipid nanoparticle platforms, which is also accompanied by a number of distinct advantages. So I'll get into a little more detail on each of these now. So what is a transposon? Transposons are DNA plasmids, or naturally occurring elements, which we historically, our founders, isolated and developed and proved on over time. A number of other groups have done this as well, but not that common in the gene therapy field currently. So DNA plasmids, which carry, as you can see in the center panel, a cargo, i.e. the gene insertion cassette, which is flanked by ITRs. The plasmid is co-delivered via electroporation in our case with a transposase via mRNA. The transposase is translating the protein in the cell. The transposase recognizes the ITRs, cuts them out of the plasmid, and inserts them into the human genome permanently, so permanent integration. That has a couple of advantages for cell and gene therapies. One, large cargo capacity, hundreds of kb, not limited as most vectors, viral vectors are, that particularly are involved in transient or permanent gene delivery and or integration. With regards to CAR T-cells, this also preferentially produces TSCM, or T-cell stem cell memory CAR T-cells, which have been associated with efficacy and safety by a number of different academic and public institutions using a number of different CAR T-cell products. For gene therapy, not only large cargo capacity being a significant advantage, again, but also permanent integration, and which is particularly relevant to diseases that affect young children, i.e. you get around liver washout over time with non-integrating vectors. And therefore, we go into, and you'll see in the future, a number of indications that are relevant to younger children with this. What is Clover? Here's a very quick diagram. As I mentioned, effectively a hybrid between the CRISPR and zinc finger or talon concepts. You see two Cas9 molecules there using a left and right guide RNA to bring together two halves of a Clo51 endonuclease to do the cutting. Advantages to this are, one, because you have a left and a right guide RNA, low to no off-target cutting. So low to no off-target indels, depending on which system you use to assess this. And activity in resting T cells, particularly important to the CAR T-cell field and particularly important to our products, as I've mentioned, where we are principally trying to generate TSCM CAR T-cells. The move from AAV to deliver transposons and Clover and transposase, to lipid nanoparticles to develop one or both of those or all three of those is something our company is particularly moving forward and successfully, as you'll see, moving forward. And that you would expect to be accompanied by a number of different advantages, particularly in terms of speed to developing a product, manufacturing, scaling, and cost of goods versus AAV vectors or other viral vectors. So I'll talk a little bit about CAR T-cells first here and where we're going in terms of a big picture with CAR T-cells. First of all, we've talked a little bit about TSCM. I'll get into that in a bit more detail in a few minutes. But we're now moving to all allogeneic products. As I think most of you know, significant advantages associated with that. And with regards to production methodologies for CAR T-cell products, proprietary booster molecules, which I can't go into a lot of detail publicly, but they are a novel platform that's utilized to increase the quantity and quality of CAR T-cells produced during manufacturing. So with regards to gene therapy, again, getting into both a large cargo capacity with transposons, moving towards novel ways of delivering transposons, as well as potentially Cas Clover, and then again, a broad number of applications, indications that this allows us to produce, approach, I should say, perhaps more effectively than most. This is our current pipeline. You can see up there that we are currently conducting clinical trials with a number of autologous CAR T cells, both in hematologic malignancies as well as solid tumors. And you can see that we are rapidly approaching the clinic with a number of allogeneic CAR T cells, both in hematologic malignancies and solid tumors. And then down at the bottom, you can see that we are moving towards the clinic with a number of classical gene therapy products targeting diseases such as Ornithine transcarbamylase deficiency and hemophilia A. Of course, there's a lot more in the pipeline beyond this that's earlier in development. A little more detail on our immuno-oncology programs, i.e. our CAR T-cell programs. This is a standard construct for PoseidaTherapeutics CAR T-cells. Because of the large capacity with the transposon, we can put multiple genes in, and this is our standard 3-gene construct, which contains, of course, a CAR, but we've actually put 4 CARs in a single product, so that would then be a 6-gene construct. We also have a safety switch gene in every product, which produces a protein which allows us to administer a small molecule drug intravenously to a patient if indicated by adverse events to eliminate the cells in vivo, as indicated, and a drug resistance gene for selection during manufacturing that allows us to deliver a pure cell therapy product, particularly a pure CAR T-cell product. You could imagine where that might decrease toxicity. I've spoken a bit about TSCM, or T stem cell memory. So why is that important? So these are the stem cells for T cells. So when you are vaccinated or exposed to a pathogen, these are the cells that remain in your body for decades afterwards and remember that antigen, so that they then, when exposed to it again, differentiate into the effector cells on the right-hand side that then do the killing. But of course, those effector cells also release cytokines, hence the source of cytokine release syndrome that is a significant issue with CAR T-cell products, and of course, all the other associated issues like ICANS, macrophage activation syndrome, et cetera. So you can imagine why having a high component, a high percentage of these cells in your product would create a safer and more efficacious product. So, as I mentioned earlier, we initiated our clinical trials a few years ago with an autologous product targeting BCMA for relapsed refractory multiple myeloma. We have been conducting, I should say, what is a fairly standard dose escalation, 3+3 design, plus dose expansion study in oncology or multiple myeloma, or even the CAR T-cell field in general. But we've added on a number of exploratory cohorts over the years to address a number of very unique hypotheses as the CAR T-cell field expands from nascent to discover more and more aspects of CAR T-cells that could be improved. Things like combinations with rituximab, combinations with Revlimid, multiple doses, as well as outpatient dosing, which has now become standard for this product. You can see that there are a large number of well-known institutions and well-known investigators that are participating in this study currently. This was funded in part by CIRM and received RMAT designation. But as I mentioned earlier, we're moving towards allogeneic CAR T cells very quickly. And as many of you know, there are significant advantages of allogeneic CAR T cells over autologous CAR T cells. First of all, made from a single donor, a single healthy donor, as opposed to each patient contributing their own T cells. Each oncology patient who has been exposed usually to multiple cytotoxic regimens. Therefore, better quality CAR T cells, better quality T cells. Each healthy donor can manufacture many, tens, hundreds of batches, or of lots of this. Doses, I should say, out of a single lot or single batch. So these can be stored, cryopreserved, sent to the site, left there ready to be administered to a patient when the patient comes to the door, i.e. off the shelf, takes out the weight logistical issues with autologous CAR T cells, plus, of course, cost of goods. So we do this in a fairly standard way. We knock out beta-2 microglobin, i.e. MHC class 1, and the TCR using Clover. We also utilize our proprietary booster molecules in the manufacture of this to generate more CAR T cells and high quality CAR T cells. As you can see in the graph here, these allogeneic CAR T cells work equally as well as autologous CAR T cells in a murine model of multiple myeloma, completely eliminate existing tumor cells in a very short period of time. As I mentioned before, we're now moving on to allogeneic CAR T cells, very similar trial design that is just about to start. This product here, BCMA-targeted allogeneic CAR T cells for multiple myeloma, just cleared the FDA, the IND just cleared the FDA, and we're about to treat a patient in the immediate future. As I also mentioned before, autologous products for solid tumors. We initiated a clinical trial for PSMA-101, PSMA-targeted CAR T cells for metastatic castrate-resistant prostate cancer about a year ago. Trial's been moving forward nicely. We actually released our first data set at the CAR-TCR meeting approximately a month and a half ago. The results were quite impressive. The first 9 patients treated, you can see that the majority of patients had decreases in their PSA. Some of those, if you go down to the right-hand side of this waterfall chart, had concordant decreases in PET uptake, SUVs as it's called, standardized uptake values, both on FDG and PSMA PETs. And as a matter of fact, some of the patients, particularly the one on the far right, near complete responses or complete responses, as I'll illustrate in a bit more detail on the next slide. So this is that patient in the far right hand of the right-hand slide. You can see on the panel on the left, this patient received the CAR T cells. You can see in blue, the CAR T cells expanded up into levels that are generally considered the efficacious range. Simultaneous to that, the PSA in red decreased down to nearly undetectable. You can see in the right-hand panel that on PSMA PET imaging, the lesion, the uptake and lesion nearly completely resolved. This is the main left-hand pelvic lesion this patient had, a metastatic tumor. And the same thing happened on the FDG PET scan on the bottom panel, and then on the far right, before and after, on the bone scans, marked improvement. Perhaps the most interesting thing is the investigator went and biopsied this tumor, which you can see is quite large, at approximately 4 weeks to 6 weeks in, and in that tumor found no residual tumor cells, but by PCR, PSMA-101 CAR T cells. So effectively a pathologic complete response accompanied by infiltration of CAR T cells demonstrated, perhaps for the first time. So where are we going next? As I said before, moving from autologous to allogeneic. So our next program to enter the clinic in upcoming months is expected to be PMEEK1C Allo1. These are allogeneic CAR T cells targeting MEEK1C, which you can see illustrated up on the left-hand side, is the tumor-selective hypoglycosylated C-terminal cleaved portion of MEEK1, which is expressed in epithelial cells broadly, so lung cells, things lining your aerodigestive tract, and therefore is expressed in most of the major solid tumors, breast cancer, lung cancer, colorectal cancer, pancreatic cancer, et cetera. And you can see that we've generated these allogeneic CAR T cells, administered them to murine models of triple-negative breast cancer and ovarian cancer, and again, seen complete elimination of the tumor. So we're quite looking forward to moving these into the clinic in the near future. Leave you with a few slides on our gene therapy programs. As I mentioned earlier, ornithine transcarbamylase deficiency, OTC deficiency, the first target. Over the years, we've moved from AAV, delivering both transposon and transposase, demonstrated levels of transduction in the liver, which would effectively cure these animals, did cure the animals, would be expected to cure patients, to lipid nanoparticle hybrids and solely lipid nanoparticles to accomplish effectively the same thing. And this demonstrates that it's gotten up to levels with those combinations in the liver with newer programs similar to what is expected. And in fact, again, this is prevented in murine models death of the animals versus controls. And lastly, our nascent factor VIII program. So this is hemophilia A. And you can see that using the transposons, transposase in factor VIII deficient animal models, we have restored, this is intravenous injection, restored factor VIII levels to normal. And we'll continue to move forward, but that's all I have to talk about today. Thank you.